Anyway, I think we can get going. Cool. Welcome everyone, and let me give a brief introduction to this talk and Elena. So let me quickly introduce Elena. So Elena is works in the cancer science and epigenetic field, and she is currently a postdoctoral research associate in ETH Zurich. She previously finished her PhD project with the 2+2 PhD scheme with the University of Liverpool and IMCB in Singapore. And she also finished her master's studies and bachelor studies also in the UK. And I think she has lots of experience living and studying in the UK as well. So if you guys have any questions, you guys want to ask her, feel free to ask in the Q&A section at the end. And in this talk, she will tell us a little bit about her PhD project, her current work, her current postdoctoral project, and she also hopefully she has some experience in living in the UK versus ETH Switzerland or Singapore. I hope this talk you guys will find this talk interesting and inspiring. And to you, Elena, thank you very much for giving us a talk here.
Yeah, sure. Let me share my screen. Okay, can you guys see the screen properly? There's nothing weird going on. Right, so yeah, hold on. Okay, so good afternoon, everyone. So firstly, I would like to thank Keynes for inviting me to give this talk, and I'm also very delighted to get to know about this journal club that is organized by people from Macau to share your knowledge in the field of biology or biomedical science, etc. It's very nice to bring everyone together, especially from our own hometown to support each other. So, hold on, why is it not moving? Okay, so firstly, I would like to give some introduction to myself. As you may already heard our conversations just now, so I have completed my education actually in Santa Rosa de Lima Secondary School in Macau, and then I moved to the UK for a foundation course in science. It is just to improve my English before I enter into uni, and afterwards I completed my bachelor in biomedicine in the University of East Anglia. And afterwards, I moved on to doing my master in cancer research in Scotland at the University of Glasgow. And basically, this master course is a taught course that has like 9 months of taught component plus 3 months of research project. So basically, I didn't think that I have enough research project to apply for research experience to apply for a PhD. So what I did is that I took on some research assistant positions to kind of gain more research experience, and then I started off applying for PhD programs. But although I did attend quite a few interviews, but I did not get any offers. So I was very, very frustrated. And I reached out to this PI that I have had interview with to ask for some feedback. And they actually told me that I didn't have enough research experience to be independent to be working in their lab for a PhD. So what I did is that then I went on to doing another master purely by research in Scotland at the University of Edinburgh. Basically, it is a pure research course without any taught components. So it was 2 research projects that I did, and it really did help me a lot. So I worked with 2 big professors and got really good reference. And then I tried to apply for PhD again during my second project, and actually, luckily enough, I did get an offer after just one application. So then I started my PhD back in 2019 at the University of Liverpool, and it is actually a joint PhD research program with A-Star Singapore. So for the first 2 years, I was with Dr. Ursula McGlegg at the University of Liverpool. And then for the final 2 years, I was with Dr. Vivek in A-Star, Singapore. And now I've completed my PhD and just started my postdoc back in March at ETH Zurich with Professor Anton Woods. So actually, for some of you who are thinking about applying for a PhD in the future, here is some experience from myself that I can give. Is that if you truly want to do a PhD, and you really need to do some homework and make some effort in your application. So basically, you should what you should do is that you should read those prospective PIs that you're applying to. You read their publication to understand their work, and if necessary, you can even cite a specific paper in your personal statement or application, or just approaching emails. And it is important that you articulate well your research interests, your experience, or even your future goals, and how their lab will align your interests so you benefit to each other. And try not to use overly broad statements so that you give an impression that you are applying for 100 position with the same CV or statements. And so you should tailor your CV for the application, and you should highlight your relevant skills. Any research experience or transferable skills is also very important, like how well you present your work, like both writing or presentation, and so all the soft skills that you have basically, and why you are a good fit for their lab. And obviously, make use of your networking. So if you're doing your research project at the moment, you should reach out to your professors and PhD student or postdoc there for your advice. And don't be shy. Just ask them to read your CV, to give you some advices. And also, it is, for me, in my case anyway, it was crucial to have strong letter of recommendations. This is how I got my PhD, really. So actually, both of my supervisors in my PhD, they knew my previous supervisors when I was doing my master. So even though science is big, but a specific field that you're working in is potentially small, that they potentially know each other quite well. So with the first-hand experience that your supervisor supervising you, they will give the most honest and your prospective supervisors would really believe in that. And for those who are still doing your undergrad and planning to pursue a PhD, then you should also spend your summer probably to gain some research experience in a lab. And for me, I actually wasn't really aware of that, and then I wasted all my summer actually going back home and enjoy the summer. So, but later on, I did have to make up for it, and also prepare for your interview with practice. That this is obviously important. So make sure that you practice your presentation well, because some of the interview you will require to give a presentation for your research projects, and also prepare for question, obviously. And yeah, try not to answer something maybe very generic. And again, make sure that you do your homework. So I think that should be the tips that I want to cover. And if you have more questions, you can ask at the end if you want to know more about like applications or specific programs that if I know of. And oh yeah, sorry. Also try to apply early and stick with the deadline, and maybe broadly as well, not just sticking to your current city, maybe expanding to different city or even different countries if you really want to get a position. So I would like to move on now to my research work. So for some of you who might not be familiar with epigenetic and how it regulates gene expression, I'm giving a brief background now on this topic. So as you know that chromosomes contains all the genetic information that is made up by DNA in all organisms. And genes are basically encoded by DNA. So here, epigenetic plays an important role for switching gene basically on and off without altering the DNA sequence itself, but by chemical modifications uh, um, um, on the chromatin. So chromatin structure is basically formed by histone molecules that arrange the DNA into a structure known as nucleosome. And this structure packs the DNA tightly, which is not favorable for any transcription machinery to take place and to regular gene expression. These histone proteins consist of histone tails with certain amino acids that can be modified by different chromatin remodelers. And with certain modification that compacts the chromatin, DNA cannot be accessed by transcriptional factors for gene activation. And in the opposite case, where chromatin can be relaxed or loosened up, so promoter or enhancer regions will be available for binding of transcription machinery for activation of genes. And this mechanism has to be tightly regulated in epicellular processes. And this regulation of this mechanism can cause huge problems such as cancer or other disorders. And this is just two classic examples to show you epigenetic modification. So for inducing a stage of open chromatin, histone acetylation is the main mechanism, and it is facilitated by histone acetyltransferases, basically transferring an acetyl group onto the histone, and this would reduce the negative charge on the histone and weaken interaction between the histone and the negatively charged DNA. And so RNA polymerase II can bind to induce transcription. And for the condensed stage of chromatin, certain histone methylation is responsible for closing up the chromatin by this classic protein complex called polychrome complex, and basically it can deposit a methyl group onto the chromatin to pack the chromatin so that transcription is off. So enough for this brief introduction. I'm moving on to sharing you some of my results, some of my main findings during my PhD. So my main project is to look into the role of a meiotic protein, SYCP1, in breast cancer. And just to give you a brief refresh on meiosis, because it's not really the mainstream in cell biology. So this cellular division process is divided into meiosis 1 and meiosis 2, and this is basically to make future sperm and eggs. And at the end of this meiosis 2, you would then get 4 genetically different gametes, and each have only half the chromosome numbers. And particularly, I would like to draw your focus on meiotic prophase 1. There is a major process known as homologous recombination or crossing over. It is basically it happens to facilitate the exchange of DNA material, and therefore you have genetic diversity in your offspring. And this requires the pairing of sister chromosome, as you can see here, a tetrastate where two chromosomes line up to each other. And for this to happen, synapsis is facilitating the formation of this synaptonymal complex, which is basically a zipper-like structure that put a chromosome together before crossing over to happen. And to this date, 8 of these proteins were identified to be involved in the building block of this cytoplasmic complex, including SYCP2 and SYCP3 as the lateral element, and then SYCE1, 2, 3, text 12, and 6 box 1 as the central element that is connected by the transverse filament SYCP1. And it is important to point out that the expression of these meiotic genes have to be very tightly controlled so that they don't express in somatic cells to interfere with normal function. So as you can see that meiotic gene expression should just be restricted to sex cells, and within somatic cells you should see none. However, with technical advances to date in transcriptomics and proteomics, it is increasingly recognized that some of these genes are actually re-expressed in cancer and driving oncogenesis. And in fact, SC protein are accurately re-expressed in cancer, and this diagram is just to show you that at least one of the SC protein is re-expressed in different cancer types. So as for the role of SC protein in cancer, by literature, out of the 8 SC protein, some of them were studied for its non-meiotic functions already in cancer cells. This paper, for example, is showing that SYCP1 impair DNA damage repair, mainly by interfere with this classic tumor suppressor protein BRCA2 to interfere it from binding from double-stranded break sites, and then they find that cancer expressing SYCP3 can be sensitized to cancer treatments such as PARP inhibitors. And furthermore, my previous lab in Liverpool, they have also identified a role of X12 when it is re-expressed in cancer. It localized to the centrosomes and causing centrosomal amplification and dysfunction. And also a more recent study also reported the role of SYCP2 in conferring resistance to DNA damage agent. So therefore, my aim actually was to understand the role of these aberrantly expressing meiotic protein. Not only that will unravel previously unknown mechanism that drive cancer oncogenesis, but also it will provide implication for developing potential therapeutic targets or identifying postdoctoral factors. For my project, I was focusing on one of the SC protein, SYCP1, and which is the transverse filament of the SC in meiosis. So it has been previously detected at transcript level in different cancers, but the role of SYCP1 in cancer biology is still largely unknown. What I have done first is to check protein expression of SYCP1 in different cancers across these different cancer cell lines with Western blot. As you can see that SYCP1 is differentially expressed in different cancer cell lines at protein level. And here we decided to take this breast cancer MCF7 cell lines for our downstream analysis. And to understand if SYCP1 also expressing on the chromatin, just like in meiosis, I have done immunofluorescence just to see where SYCP1 is localized to, and as you can see that SYCP1 is actually in the nucleus. So that gives us hope, and so then we can move ahead to do some chromatin analysis to see if SYCP1 is actually binding to chromatin and what it does when it is binding. So for some of you who might not know about chromatin assays, so I have performed chip sequencing analysis, which stands for chromatin immunoprecipitation. Basically, it is an assay to identify a genomic region that is bound by your protein of interest. So how it works is that you have the cells that containing proteins that is binding to different different DNA region in the genome. And then you want to keep these in the native form. So then before you lyse the cells, you chemical cross-link it so that it stay in this native form. And then you can shear or fragment the DNA along with the bound protein of interest. Here is SYCP1 in this case. And then what I did is that I used a S1CP1 antibody to immunoprecipitate this protein DNA complex, and then the DNA can be eluted from the protein, and this DNA can be analyzed by next generation sequencing, and then you can map this read onto the genome in order to know where SYCP1 is binding to. So here, this pie chart is showing you that we did identify some SYCP1 binding peaks, and as you can see, that majority of it is actually on the promoter. This strongly suggesting that SYCP1 has a role in transcriptional control on gene promoter. So we want to know what gene promoter it is controlling. So then we take this promoter for gene ontology analysis, pathway analysis. And as you can see that SYCP1 bound genes are associated with chromatin remodeling, DNA damage response, or cell cycle, etc. And also to understand if SYCP1 is important for breast cancer. What I did is that I knocked out SYCP1 using CRISPR-Cas9 in this MTS7 breast cancer line. Basically, what I did is to target SYCP1 genes just after the start codon of the gene, and then I transfected the CRISPR machinery, including the Cas9 protein and the guide RNA, into the cells. So basically, what it does is that this Cas9 protein will cut where guide RNA is targeting, and this is an error-prone process. So when the DNA tries to repair itself, it would normally induce a insertion or deletion, so that it could cause a frame shift in the gene, and it couldn't make a functional protein. And then after transfecting into the cells, then you have a bulk edited population, which is not homologous, but we want to have a homologous population that doesn't have any weird editing events, and we need to make sure that SYCP1 protein is gone. So then what I did is that I single cell plate attempt for clonal expansion. So then after I have grown enough number of the cells, I can then take protein and genomic DNA for genotyping. So here I use topocloning. Basically, long story short, that you can screen each single event using single sequencing. This is just two examples to show you that I have managed to induce a single base pair insertion, some with 9 base pair in deletion, and both cases cause a frame shift. And Western plot also showed that SYCP1 protein is indeed gone. So next, I want to use this SYCP1 knockout cells to understand how it affects cells by performing RNA-seq analysis. So with SYCP1 knockout, we have identified a number of differential expressed genes. So here we focus on the downregulated genes, and to understand how these genes are regulated directly by SYCP1 binding, I overlap these downregulated genes with the SYCP1 peaks that I have identified from chip sequencing. And then I have taken these genes for pathway analysis, and consistently you can see that these pathways are have shown up, and this is just some genome browser check to show you some key regulator for cell cycle and DNA damage that is regulated directly by SYCP1. And I also did some functional assays on SYCP1 knockout cells. So as you can see that SYCP1 knockout has reduced cell proliferation and also chromatin and also colony formation ability. So altogether, it suggested that meiotic SYCP1 protein, it also binds to chromatin even when it re-expressed in somatic cells, and that it regulates cell proliferation by directly binding to the promoter of these genes that are responsible for this function and potentially acting as a transcriptional activator to promote breast cancer proliferation. And actually, I have also taken on an additional project during my time in Singapore, which is to look into a non-canonical role of metabolic enzyme ACSS3 in driving breast cancer cell migration. So this project actually based on a previous project that has already been published in the lab. So they were investigating the role of a transcription pulsing factor NELF-E in regulating a process known as epithelial to mesenchymal transition in breast cancer. So EMT, epithelial to mesenchymal transition, is a process where cancer cells basically lose their epithelial structure and acquire mesenchymal properties to make the cells much more invasive for metastasis. And that enables them to detach from the primary tumor sites to metastasize or spread to distal organs. And what they did is that they used breast cancer model that they can induce EMT by basically by overexpressing these transcription factors LUT and SOX9, and their expression can be induced by doxycycline treatment. So then they did a chromatin mass spec analysis, Q+1, basically to look into NELF-E protein interactor on the chromatin. Basically, they identify a bunch of protein interactor, but interestingly, they find ACSS3 as a significant hit. And why is this interesting to look into? Because ACSS3 is not known to be localized to the nucleus. It is a metabolic enzyme and is normally found to be expressed in the mitochondria. And in fact, it is increasingly known that cancer can harness metabolic pathways for epigenetic regulations to drive oncogenesis. And more study reported that the intermediate metabolites that drive chromatin dynamics through post-translational modifications to alter chromatin structure and function. And for example, SAM here is an intermediate metabolite that is derived from TTA cycles in the mitochondria, but it also is a known substrate for DNA and histone methyltransferase for gene expression. And similarly for other metabolic intermediates and enzymes to regulate epigenetic modification. And just to give a brief introduction to ACSS protein, it is basically a class of protein that is known to be catalyzing acetyl-CoA from acetate as a substrate or other substrates. And it is mainly for lipogenesis and expressing in mitochondria. And for ACSS3, it's basically enriched in the liver. And ACSS3 has also been reported to have roles in cancer progression in gastric cancer and also in bladder cancers. And because it was actually detected as an interactor, as I mentioned, on the chromatin, and we hypothesized that ACSS3 may be localizing to chromatin to convert acetate for acetyl group for histone acetylation to induce gene activation. So to start looking into ACSS3, I have run a Western blot across different breast cancer cell lines, and as you can see here, compared to the non-tumor reagenic MCF10A, you don't see expression here compared to other breast cancer cell lines. And just to mention that breast cancer, just like other cancers, is classified into different disease subtypes. And here, for breast cancer, we have 5 main subtypes. And each subtype, they manifest different prognostics or clinical outcomes. And here, I would like to draw your attention on triple-negative breast cancer, which is actually the most aggressive breast cancer disease subtype. And here, I use MDA and B231, and is lung metastatic subline LM2, and also the EMT model that I have mentioned to you previous slides, a couple of previous slides. And as you can see that in the metastatic line and also in the EMT-induced line, you can see an upregulation of ACSS3. So this implies that ACSS3 implies that this EMT or metastasis may be depending on ACSS3. And to look into if ACSS3 is actually affecting cell migration, so I knocked down ACSS3 using siRNA and shRNA. And as you can see here, by transwell migration assay, you can see significant reduction in migration ability with ACSS3 silencing. And also, I've done transcriptomic analysis to see what genes are affected by ACSS3 knockdown, and consistently you see EMT or angiogenesis. This pathway are related to metastasis. They are both downregulated in both the models. Sorry. And so then I move on to studying the mechanistic role of the mechanistic role of ACSS3. So basically, we only have results from the mass spec data, and so we had to validate whether it is actually binding on chromatin. So I did a simple chromatin fractionation assays. Basically, how it works is that you have the cells that you can lyse with buffer, a rather harsh buffer that would lyse everything, but not the chromatin fraction. So then you separate this fraction with centrifugation, and then you have both the fraction to be loaded on the Western gel. And indeed, I would like to draw your attention to this last lane. You see that even though you have a high amount of ACSS3 in the soluble fraction, well, because it is on the in the mitochondria as well, but you also see some expression on the chromatin fraction. So that gives us confidence to also do chromatin analysis as well. But in this case, I did cut and tag instead of chip sequencing because ChIP-seq didn't work for this protein, but it basically serves the same purpose. So as you can see that ACSS3 is also enriched in the promoter, but also in the enhancer region. And then these are the metaplot and some genome check to show you that ACSS3 is enriched around the transcription start sites. So so we want to know how exactly ACSS3 is regulating gene expression as well. And as I have give some introduction to histone modification, we basically have this hypothesis that the role of ACSS protein, because it can catalyze acetate to acetyl-CoA. So if ACSS3 is actually on the chromatin, could it be generating acetyl group locally on the chromatin and deposit them on histone by working with histone acetyltransferase? So to test this hypothesis, basically, I've chosen HPK27 acetylation as one of the most abundant histone modification. As you can see that it was downregulated globally by ACSS3 knockdown. And so to test if it works with any histone acetyltransferase, I started off with one of the most abundant histone acetyltransferase, P300. And so I went chip sequencing on both P300 and H3K27 acetylation. And then I overlapped these peaks with the one I find with ACSS3. And indeed, we actually get quite a good number of co-bound peaks by the three peak analysis. And then we overlap these peaks with, just like how I did with SYCP1, I overlap with the downregulated genes that we identified from RNA-seq analysis. And we want to understand what this acid of ACSS3, P300, K27 acetylation is regulating. So we also take this subset of genes for pathway analysis, and you can see that this pathway are associated with cell cycle basically related to invasiveness like GTP-based epithelial remodeling and all this kind of pathway, and again some genome check to show you examples of candidate genes that are important for EMT functions that you can see that is enriched with ACSS3, P300, and K27 acetylation. And this suggests a direct regulation of this gene by ACSS3 binding. And so to summarize for this part, I have shown you that ACSS3 phenotypically it is important for breast cancer cell migration, and mechanistically it goes to the chromatin and regulate this gene directly through P300 and H3K27 acetylation. So just to switch gear a little bit, I'm going to give you, for the next 3 slides, a brief introduction to my current project that I have just started a few months back. And this has nothing to do with cancer biology anymore. So my current project is to explore and improve an in vitro oogenesis system by looking into meiosis. So we use mouse here as a model, and to improve or develop an in vitro system, it is important to understand what happened in vivo in order to know how in vitros work, basically. So this is the whole timeline of a mouse development, particularly in for sex cells. So basically, after mature oocyte fused with a sperm, right, you get embryonic, and you get an embryo. And at this early stage, it is called a zygote. So a zygote can be divided into 2 cells, 4 cells, and so on until it reaches to a state, to a more specified state where 3 germs layer are derived. So here I would focus on the mesoderms, where the precursors of germ cells occurs. And this is known as the primordial germ cells. And for the next few days of the embryonic development, these primordial germ cells, PGCs, are migrating to the gonads. So what gonads mean is the embryonic sexual organ, which will become, for female is ovary, and for male is the testis. And then at this stage, and this embryonic state, 12.5, it is when sex is determined. So PGCs will become oocyte in the XX cells, and then in XY cell it becomes spermatocytes. So for female, meiosis occurs before birth, but it actually get into a dormancy after birth and until puberty, then meiosis resume, and every month, or in mouse is different, then you get a mature oocyte. And for males, meiosis wouldn't happen until puberty. So for my project, I would be focusing on the female trajectory. And this work is actually based on two really important Japanese pioneers, where they use embryonic stem cells or induced pluripotent stem cells as the starting point to induce stage by stage from epiblast-like cell, primordial germ cell-like cells, all the way through entering meiosis, primary follicles, and then mature oocytes. And they have even managed to use this system to generate oocyte to put and then fuse with sperm by in vitro fertilization and put back into our surrogate mouse to give birth to viable pups. So this work is very impressive, but it does have key limitations. Because the efficiency is extremely low with a lot of starting material, you only get 3% a successful rate of getting this result. And then they pointed out that these oocytes actually have more fragile cytoplasmic membrane. They have lower copies of mitochondrial DNAs and aberrant gene expression in different stages, and obviously when you have bad gene expression, then you cannot make protein in time. So then synapses couldn't happen properly, and eventually high rate of aneuploidy. So the aim of my project now is to look into this and try to improve this in vitro system from the angle of meiosis, and particularly through the entry of meiosis and in prophase 1. So to understand these processes, including how transcriptional control is regulated for precise timing of expression of meiotic genes, is important to find clue to improve this system. And why is it necessary to develop an in vitro system for that? Because stem cells material is normally very difficult to have access to because of also of problems like ethical evaluations. And so if we develop an in vitro tool, then we can have a system to study development or meiosis for disorder or infertility. And with these tools, it is easier for us to do molecular analysis and easily like knocking out a gene or do mass spec DNA sequencing with a higher amount of starting material. So, potentially, this system could even generate oocyte even in other mammalian species. So yeah, that's it for my science talk. And here I would just like to show you some memory during my PhD. It's a bit of a shame that I don't have much picture when I was in Liverpool because I was living in this COVID time. But then in Singapore, I did have a really good time with my lab. We have a lot of outing, and I have to say, working or studying in Singapore, I think I will never regret this choice. It is a very nice place to stay. If you are a foodie like me, we constantly have a lot of restaurant outing and stuff after living in the UK for so many years, especially. And they are also very supportive lab mates as well, and I cannot thank them enough. And also, during this Iraq program, I got to meet also other students from other UK universities in different fields of science and engineering, and some of them have even become my long-term friends, and I've also made some really useful networking. And I actually knew King through this guy, Dimitris, which is who is a fellow PhD student, and this is actually his farewell party before he left Singapore. And last but not least, I would like to thank both my PhD supervisors, Viwei and Uller, and also all the lab members in their lab, and my current supervisor, Anton Lutz, and thank you all for listening. And I'm happy to take questions and have a discussion if you have any.
Ooh, thank you very much for the talk. Any questions? I guess we can start with the scientific questions in the talk, and then we can move on to ask Elena any questions about, life and in the UK versus Switzerland, etc.
Yeah. Any question?
Yeah, cool. Yeah, do you guys have any questions? There's a question in the chat, but yeah.
Yeah, okay. I can stop sharing. It's yeah. So for the doxycycline, is it to build up resistance to allow extra expression of the following genes? So yeah. So basically, it is not really for building up resistance, but because this so this overexpressed genes is in a vector where it is downstream of a specific promoter that can only be bind by doxycycline so that it overexpressed. So without really treating the cells with doxycycline, these genes they don't get overexpressed. So it's basically just induction system and inducible system actually. And this is useful for some cases where you want to compare induced or non-induced stage for comparative analysis, if that answers your question. You're welcome. It's also okay for you guys to turn on your microphone to speak to me also, and I would like to get to know you guys as well. And there's no stupid question really. Like, please ask anything.
Yeah, Ashley, do you have any questions? I guess this is the most similar to your field, Ashley. If you have any questions, feel free to ask.
And if you want to understand what it is like to, do a PhD in Singapore, or how is it like living there, yeah.
Okay, I think there are many people typing in the chat. But yeah, so I can share some of my kind of feedback. So I do computational chemistry only, so I don't really know any of the techniques, but it sounds very impressive. So I can understand, the knockout and the interaction. I am quite impressed that you can find which protein are actually interacting with the POI, for example, is quite cool. And then you can do this mass spec, etc. And it's quite logical, and I can kind of understand it. But I guess for those of you who have a more biochemistry background, might be able to understand more easily. But yeah, it seems quite impressive work. Thank you, Elena. And yeah.
Oh, thank you. But yeah, I try to make it easier for everyone to understand, just to explain different knowledge. But it is indeed a beautiful thing working on epigenetics. You really get to use a lot of different techniques to find your research question. No, it's okay. It's okay. Yeah, no worry. I can read. So you would like to know more about researching in Singapore. What would you say is great there compared to the UK or elsewhere? So researching in Singapore, I would say it is definitely different from the UK. So in Singapore, at least where I was in in my institute, so basically, well, they I would say they're very organized, and funding is very competitive, and they do have some main scheme that they want to focus on, and for now the mainstream in Singapore, like in A-Star, I don't know about other university actually, but they are focusing more on translational medicines like cancer, diabetes, and now they also focusing mainly on aging as well. And so they are good for looking for drug targets and using patient samples and a lot of resources that you can get access to, really. So this is very convenient. But one con I would say is that because they're focusing on advancing medicine, it means that they would spend less resources in looking into basic biology. Unlike in the UK, when you really want to study what certain protein does, how it flow, and like what a specific organelle is doing. In the UK, is actually better for that. They actually have more curiosity for that, I would say. And elsewhere, and actually researching in Zurich, I cannot give you too much insights yet because I've just started as well. But I would say it's basically quite similar to the UK actually. The only thing in Switzerland is that because Switzerland is really rich, so they have really huge funding in ETH. Like the PI I'm working with now, he didn't even ask me to write a grant as a postdoc basically, and he just has all this money to run the lab. So this is a very good thing to do research in Switzerland, because as your boss is stressing about money, they stress you out as well. So I'm not saying that without stress is good, but at least it doesn't create a very toxic relationship. Then you can kind of really focusing on like doing your project, and then he will give you all the resources, etc. So yeah, it's definitely worth doing science elsewhere as well. Okay, with Ashley, thank you. Would you think integrating AI in those research and is becoming necessary? Yeah, yeah, I would say yes. Obviously, everyone is going into that direction, right? So depends on what AI that you're using. I mean, even just with ChatGPT, it's already helping you a lot. And there is actually some analysis you can actually just upload on ChatGPT, and it will give you a very accurate answer sometimes if you ask the question correctly also. But as for which specific type of AI, I would say, because for my future project, it is quite important to understand like how genes is regulated in the system that I'm using compared to what is actually happening in vivo. And there's actually a lot of work has already been done in vivo as well that we can compare to make this too. And that would require quite a lot of bioinformatics. And I think I will only get to this point where I can use all the developed pipelines for analyzing data, but this could involve some AI tools as well, I suppose. Yeah, I'm sure. Yeah. Okay. If that answers your question, and if you need to ask any further question. Yeah, by the way, yeah, I'm sure food everywhere else is better, but not actually in Switzerland. I would say Switzerland actually has worse food than the UK. Unfortunately, because you don't really see, well, you do see a lot of Chinese people around, but it's not as much as you can see in the UK, right? That now everywhere with Chinese restaurant, Chinese grocery stores, and but in Switzerland it's quite different in that sense because in Chinese restaurant they really make food to impress Swiss people. So if you know what I mean.
Yeah, follow up on that, can I ask about would you mind sharing some of your experience in, studying slash living in the UK versus Singapore versus ETH Zurich? Like which one is your favorite, etc. Like how do you find it from moving to UK to Singapore to ETH, etc. That would be very interesting. Thank you.
Yeah, sure. Yeah. Um, yeah, in the UK, sometimes I do miss living in the UK. It's just it's to do with the language actually, because in now that I'm in Switzerland and their official language in Zurich is German. So I mean it's okay not to speak German in Zurich because it's quite international as well. But in the UK you don't have this problem to worry about. Yeah, I love being in the UK actually, but the the thing is that um, the UK economy is going worse and worse. So I don't really see how I survive well in the UK, at least for the next 10 years. So I think it's time to move on to if I have other opportunity elsewhere. And the reason why also the reason why I chose to do this PhD program is very appealing to me, like half in the UK and half in Singapore, because we're all from Macau and going home is so easy. So yeah, it's like 3 hours I would be home compared to when I was in the UK. I really need to plan very well when I get to go home. And um well yeah, but actually unfortunately when I was in the UK, COVID happened, and then I didn't manage to go home, and then I was kind of stuck in the country. And even when I moved to Singapore, I was still stuck in Singapore because COVID didn't come to an end yet. Um, um, and so I actually didn't get to experience much for the first few months in Singapore because it is like extremely strict, like 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 I remember I arrived there with the rule of two. Basically, you can meet one other person at that time when I arrived there. And then yeah, I didn't get to explore much, but very soon they lift off all the restrictions, which was great. And then I got to you know go out and get to know people from my same program. And yeah, then I started enjoying Singapore a bit more. But yeah, I mean, between living in the UK and living in Singapore, I would say because Singapore, it doesn't really have winter. It doesn't really have any season. So this is a bit boring if you're okay with that. Otherwise, Singapore is a great place because me, I cannot deal with extremely hot weather all year around. Um, and apart from that, food is nice, but I would say Singapore is a very expensive city. So, but they do give you enough scholarship though, if they're much more generous compared to in the UK. I received really quite tight stipend when I was in the UK. I don't know for those of you in London, it could even be worse. Um, but yeah, in Singapore, the most expensive thing is probably housing So I spend like at least half of my stipend just to rent a place, living with 5 other people in the same flat, just to be close to work, really, because I yeah, because I don't want to spend so much time commuting, but then it costs me so much. But then they have a lot of hawker centers, like food court. That's how they call hawker centers. That they have all kind of local food for very cheap price. So this really helped my life a bit. And if you have ever been to Singapore, you would know that every street block you will have a hawker center for really cheap food. So that's very nice. Um, coming to Switzerland in Zurich, it is like if I mean there are good and bad, right? Every place have its own good and bad. And in Switzerland it is even more expensive than in Singapore because you cannot find any hawker centers. So housing is expensive. Food is yeah, you don't really want to eat out in a restaurant, really. You want to cook every meal by yourself to save that money. Um, um, but they do give you very good scholarship and really good salary if you do it. If you're not a PhD student, so you definitely can save up if like I say if you cook yourself. And and on top of that, you have really nice mountains, very nice lake view, and if you're a bit of an adventurer, you can always go to different places to explore when you're in Switzerland. And it is in the central on Switzerland, meaning that you can fly everywhere in Europe very easily. Um, so yeah, I'm still exploring Switzerland at the moment. But I'm enjoying it so far, I would say. If you want to know something more specific in that I haven't covered.
Ooh. What is your next city, or are you still exploring Switzerland?
I think I'm planning to settle down now because also that's the reason why I choose Switzerland as well, because Switzerland is very safe and economically it is actually a safe ground in Europe, as you may know that how unstable it is, everywhere now. But Switzerland is rather doing quite well actually. So to settle down here, I think it's rather more promising compared to other European countries, I would say. But I love Europe, so in long term, I think I would be staying in Switzerland as well. But then, like I said, Singapore is also a good choice if you prefer Asia. It can be a little boring if you live there for long enough because it's just a small city as well, and which is why Singaporean during holiday is in Singapore is very quiet during holiday time because they all go out on holiday, and and also it gives easy access to other Southeast Asian countries as well. In Singapore.
Cool. Just out of curiosity, what is your career plan? Like you mentioned, you want to
Yeah,
stay in Switzerland, right? Yeah, just
yeah, yeah.
curious.
Yeah, this is a very important question, actually. Long-term career plan.
Like, do you
Like
tend to work
even
in
I would,
industry or academia, those kind of thing? Like, yeah, that's just
yeah,
out of curiosity. I don't know.
I will give you a very honest answer that if I do get a chance leaving academia, I would just say bye to academia with no hesitation, really. To be fair, because I also want to try different things as well, and although doing a PhD can be fun and also hard, but and I also got a lot out of it. But during this whole PhD journey, I really realized that academia is really not my thing. Like because that's all they depend on is grants and papers. And and for me, I see a lot of like you know like like how should I put that? but it's just really not my thing to spend all my life and energy into making a paper, if that makes sense. If it sounds very demotivated, I'm so sorry, but then I need to say doing a PhD is still fun to answer a research question. But for me, I also want to try different things as well to see how I can use my knowledge to apply to industry and learn more new stuff and stuff that you cannot learn doing academic. So yeah, but then as I was trying to find a industrial job in Europe, I really got no luck, especially in Switzerland, where I'm only restricted to Zurich and Basel really because these two cities are more international, so you don't have to speak German. But I think I applied for over 50. I didn't get a single interview. So it is tough. Job market is tough right now. So, but then, like I coming out of a PhD, you have a very niche skill set. So when you actually apply to do a postdoc, surprisingly, it's much easier for me. So in this current lab I'm in now, this is my second approaching email that I've sent, and then I heard it back basically because my supervisor he likes the fact that I have some background in meiosis and we can just be a match really. So I will see how I'm doing at ETH before before I'm trying to apply for an industrial position again.
Cool. What type of industry are you interested in? Just for curiosity.
A smaller biotech company, I think, and maybe like drug development, clinical trials, and even I want to try project management. So that would be something that I can work on during my current job that I can get more like maybe qualification or other skill sets. Yeah. So see how it goes really. And there
Cool.
is actually a lot of startup company in Basel as well. They do all these different biotech company on just next generation sequencing, some other like stem cells company, organoid stuff, and which is particularly useful for well with my current project because I'm essentially doing a 3D culture now, so this is also the mainstream as well in cell culture.
Cool. I know you mentioned you want to stay in Switzerland. I'm curious about, what is the requirement for example, like work visa or like how do you get a residence permit or something like that in Switzerland? Just for curiosity.
You need to get a job first in order to get the permit. But unfortunately, I'm not sure about you guys, but I took advantage of my Macau citizenship because I have a Portuguese passport. So so as a EU national, well, I think actually now because Switzerland is actually not part of the EU. So actually, anyway, with me, I can stay 3 months in Switzerland without any problem, without any visa. But I think people with passport from Macau can also stay. So what I did is that I stay in Switzerland for a bit to look for a job so that if there's any interview, then I can attend in person as well. And yeah, it really depends on it really depends on your employee really. If they're okay to recruit you, then they give you the work permit, then happy days. But yeah, but generally it's probably quite difficult to find a job. I'm not sure about other nationalities really outside of the EU. But what I know is that in ETH, it is worth pointing out actually they are actually restricting Chinese people, Russian people, and people from Middle East to come to do research is because of this sensitive thing that you might have known of, but in order for them to come to do research, they actually have to go through a lot of background checking. So it's a lot of paperwork, and it's really not easy it's actually making it difficult for them to come over to Switzerland to work now. But otherwise, I think if you have the right qualifications and good enough experience, then it should be okay. But actually, there's one thing I need to mention when I was struggling to find an industrial job. So I tried to make some networking, through LinkedIn and also through people I know. They all told me that networking is really the key. So, in this big company like Roche or Novartis, they have like thousands and thousands of applications that they receive every day. Then the HR, they basically just won't go through that. But then if you know someone in the company itself, they can actually refer your CV to this HR. So you bypass all the applicants basically. So I guess it could be a tip actually, but yeah. So get to know people. Networking is always beneficial, really.
Ooh, any other questions? I actually want to ask, do you have any advice for us, like young scientists, any challenges you face and, any advice in general? That would be very helpful.
In terms of life or research or study,
Like
in terms
anything,
of
anything.
anything. I don't know. It gives
Oh.
me a lot of flashback, really. But I'm trying to think if there's anything I can. I mean, if any of you actually really wants to do a PhD, if you're still in your undergrad or master stage, then I would say, yeah, I mean, if you're committed, don't give up. You will face a lot of failure and application, really. It's just like what I did. And I did not really have a particularly good academic background as well. I just kept trying. Whatever I don't have enough, I try to make it enough. And yeah, same mentality really when I was doing a PhD. It wasn't really easy for my PhD, to be honest. It's not just the research, but also the relationship with my supervisor as well. So the benefits of doing this 2+2 is that I got to experience more labs and experience more places. But then there is also a big con to this, is that the two supervisors, they might not have the same idea, and that they both have their own ideas, and sometimes they don't agree with each other, and you're kind of just in the middle. And that actually, this experience actually taught me a very big lesson. As I don't know, it depends on people how you will deal with this kind of situation, but I would say be resilient and, don't be afraid, and speak up when you have any idea. And if you have anything that you don't agree with, yeah, I would say yes, they have authority over you for sure, but then you also have your right to speak up as well, and don't really let. I mean, as you are accumulating more knowledge and more experience, you can actually gain more confidence in what you're doing. So sometimes your supervisor can be wrong, and it's okay to say that. And yeah, this is just this is just one aspect to make you more resilient and deal with different situations. For me, coming out of a PhD or master's, or it's not only I'm not only taking away with experience in research or knowledge, but also how how do you I grew up as a person, let's say, then you really feel that how much more mature you are after you have been through such a tough situation, basically. Um, and so yeah, I would say like for now, if you're applying, keep improving, and and do read a lot. I would say read a lot and keep accumulating your knowledge. One day it will be useful. I'm I'm sure for that. And yeah, don't be afraid to explore as well. And yeah, yeah.
Cool. Thank you very much for the advice. Any other last questions?
Yeah, also if you have any conference opportunity, go to conference. And all the networking is very important. And for me, I miss all this chance to do my networking because I constantly try to rush for data and stuff, but I also understood that there's no end to it, basically. You always have data to get, but you can also spend some time on something also very useful as well. Yeah, but otherwise, I can only say good luck to you guys. Yeah, doing science is fun.
Cool. I was reading my notes just now. I actually have kind of questions about the project now. So, the first two projects is kind of cancer related, which is, it's
Mhm.
relatively easy to understand. And I think I can understood most of them. And I think I get kind of confused in the postdoc project. So it seems to be completely different than a cancer field. Like, is it completely different, techniques and, direction, project, etc.?
Yeah. As it is very different, indeed. But sorry, I didn't explain more. I don't want to drag the talk for like forever long. But so for this project, like I mentioned, I'm using a in vitro oogenesis system. So it is basically a culturing system, which lasts for 45 days long, really. And basically so for my stage, I am getting used to just using this system because I've done a lot of cell culture for cancer projects, but it is never as close as to how complicated this culture system is. It requires a lot of different culturing methods, like there's a 3D culturing system, and it involves a lot of cytokines to differentiate the cells from stem cells, which is the most pluripotent stage that can become any kind of cells really. So then for certain signal include like I adding different cytokines that and then it can alter, the stem cells itself with signaling or on the level of epigenetics to make these cells more plasticity to go into different trajectory. Um, so here I'm using the stem cells, adding certain cytokines so they become like a precursor stage of the germ cell, which I've mentioned is epiblast-like cells. And then from these cells, you use different culture methods and add different molecules so that it becomes a precursor of a germ cell. And then these cells have the potential to become either a future sperm or a future egg. So I'm focusing making it to be a oocyte, so female egg basically. And then the good thing about this system is that the Japanese scientists, they developed this reporter cell line. So from the embryonic stem cell that they given us, it has like a reporter genes that fused with some developmental genes, so you can trace the trajectory of the differentiation. So what I mean by reporter is that you can actually put like a fluorescent marker to a certain gene in the cells, so that when it is expressing, the cells will shine a color. You can look it in the microscope. Basically, when you differentiate the cells in different stage, and then you can literally visually see how these cells are differentiating to which stage already. And then through each point, you are changing the system method. You're even adding, even getting into 3D culture system where you actually put somatic cells around these germ cells, because in the ovary or in the testis, it's not just really your sperm or egg is hanging around in the organ, right? It has some surrounding a microenvironment for supporting your sex cells to grow. So what we did currently is still limited to getting these somatic cells from a mouse. So we need to dissect our mouse to get into the embryo, to get into the sex organ, to take these cells out basically, and then we can culture them with the differentiated cells that I mentioned to you before, and so it will go into a follicle, and then after days, when you like adding different stuff to it, it will become a mature egg basically. But like I said, the goal of this project, because this system is kind of optimized and kind of work, but then each step, it costs so huge amount of money, and all these cytokines and everything is extremely expensive. We are talking about one aliquot of the BMP4 that we use normally is costing 6,000 Swiss franc. So just this is already costing so much money, and then eventually gives you a very low efficient outcome. So we need to find out the problem to boost this system basically, so then it can be applied to human, and also the direction in Japan, they actually expanded it to even different species to try to save nearly extinct animal back to life, or that kind of stuff. Yeah, so it's actually pretty cool. I wanted to do stem cell biology from a long time ago, but so it is a good opportunity to me actually. But and it's fascinating because yeah, you do see transformation of the cancer cells, like from a primary tumor to become a metastatic tumor, but that's all you can see really. But with stem cells, you can literally differentiate them to all these kind of cells, and there's a lot of mechanism that you can look into, especially for epigenetics. It is the ground stage of differentiation and cell plasticity, basically. So yeah, this is just the very beginning of the project.
Ooh, thank you. Any last questions?
Yeah, if you guys come to Switzerland, we can stay in contact.
Oh, cool. Yes. Cool. If not, yeah, thank you very much, Elena, for giving us the talk and sharing your experience and advice. I'm sure many of us found the talk helpful. And yeah, yeah, nice to meet you, Elena, and yeah, thank you very much again.
Yeah, it's very nice. Just keep this group growing. It's nice to see,
Okay,
you know, people from Macau coming together doing the similar things.
cool.
Yep, you too. Thank you.
Yep. Have a nice evening, everyone. And yeah, bye for now. Bye.
Have a nice weekend.